Welcome to the Ticktective podcast and video series, a program of the Bay Area Lyme Foundation, where our mission is to make Lyme disease easy to diagnose and simple to cure. I'm your host, Leah Gertner, Director of Education and Outreach. Lyme disease is the most common vector-borne disease in the USA and is a global issue. This show includes interviews with the researchers we fund, as well as other interesting people in the Lyme scientific community. We are a nonprofit foundation based in Silicon Valley. Thanks to a generous grant that covers all of our overhead, 100% of all donations go directly to support research and prevention programs. You can find out more or donate at bayarealyme.org. Today, I'm joined by Dr. Rogovskyy, who is one of our 2020 Emerging Leader Award winners at the Bay Area Lyme Foundation. Congratulations. Dr. Rogovskyy is currently an assistant professor of veterinary pathobiology at the College of Veterinary Medicine and Biomedical Sciences at Texas A&M University. But I just wanted to ask him to jump right into ticks in the nuclear zone of Chernobyl.
Yeah, sure. So the idea is to see if there's any differences in the prevalence of selected pathogens or actually microbiome in ticks from Chernobyl compared to the other regions of Ukraine. And the rationale is very simple, straightforward. In April 2016, it was 30 years since the disaster happened. So 30 years of ionizing radiation exposure, I think it's enough to see some changes in the microbial composition of ticks. So with that in mind, I went to Kiev, got permissions beforehand. I have collaborators there in Kiev, and we went to Chernobyl to collect the ticks. We spent an entire month collecting ticks, both in Kiev as a control area, which is about 2 hours away from Chernobyl exclusion zone, and in Chernobyl. So we were collecting Dermacentra reticulatis and Ixodes ricinus ticks. As on a side note, for those who are not aware, Ixodes, they're responsible for transmitting Lyme disease. So we have performed microbiome analysis on individual ticks, total of 400 something, and we actually about to publish some of that work this year, but I don't want to yet tell you what's going on. I think it's fascinating, and I want the viewers of this interview just to go and find us on PubMed and read it.
Right. I've seen one article online, maybe it was preliminary data, that was fascinating.
Yes, we published, I think last year, on we showed some difference between the prevalence of zoonotic agents in ticks from Chernobyl versus Kiev, but now we want to release the data on microbiome studies, which is more in-depth analysis of microbial composition of these ticks.
So I can talk about what I read online, which is that the rates of Borrelia infections were higher in the Chernobyl exclusion zone. I know people think a nuclear zone would not have abundant ecology, but I've seen some nuclear areas that are actually quite abundant in ecology because people are not building there and they're not going in there. So is that how it was in Chernobyl?
Right, because the area has been abandoned for 30 years. That's what has an impact on the ecology of selected zoonotic tick-borne pathogens, and or because of the ionizing radiation that the area has been exposed for 30 years. You know, it's a preliminary data that was published. It's really hard to connect the dots between the irradiation and the high prevalence of this Lyme disease pathogens in those ticks. We can only say about association, but causality is very hard to determine. So more studies needs to be done. That's why we decided to do microbiome studies to dig deeper into this area.
And did you have to wear nuclear protection?
Yes, the routine was quite unusual for us. Yes, first of all, we had to have a permit because there's two zones there. It's a 30-kilometer zone around the hotspot and the 10-kilometer. And you have checkpoints. You need to show the permits, and they have to be just for 1 day. And it takes time to get those permits. Also, we had to have some clothes that we were disposed after we're done for 1 day. So then taking the showers, we were checked for if we were exposed when exiting the zones. So it was quite an unusual routine that we had at that time, for sure.
And do the ticks need to be handled in a special way?
That's a good question. So we actually were measuring the irradiation level when we were collecting ticks, and we were trying not to get in those hotspots. And those hotspots are actually the area where they buried the used equipment that they were using when they were cleaning the area during the disaster 30 years ago. So if we try to avoid those spots, because the radiation levels there would exceed the 10, 20, 30, 100 times normal. So we try not to collect ticks from there.
I think that's wise. I think being a researcher in the world of ticks and Borrelia already feels really risky and brave. I mean, you're working with ticks every day. So does your lab actually have tick rearing capacity and Borrelia rearing capacity? What is, if you don't mind describing what your day-to-day life is like there?
I've been running my own lab for 5.5 years. We do different types of work. We do molecular work. We generate mutants. We do immunology work, ELISA, Western blots. We are capable of, I was trained and I had experience of working with ticks. Currently, we don't have any ticks in the lab, but we are allowed to work with ticks here in the lab. So we're trying to diversify our area of expertise.
Great. And so for the project that you'll be working on for the Emerging Leader Award grant for the Bay Area Lyme Foundation, I know that that's a diagnostic test and we won't go into it because it's probably top secret, but you will be using biobank samples, right, from the Lyme disease biobank. So you'll be using human blood from Lyme patients and you'll be testing your diagnostic on those blood samples. Is that right?
It is correct. It's one of the specific aims. We actually want to do some studies using the well-characterized mouse model, C3H mice, but by using that mouse model, we already have promising preliminary data. But at the same time, we want to shift to our work towards analyzing the human samples, of blood in particular, and we're going to use the biobank samples. I think this is a very unique opportunity to use these samples because they are well-characterized, so you know exactly if the patient was confirmed LD, a Lyme disease case, or not. So we're going to validate basically our approach by using human specimens. Yes, that's correct.
Great. I'm so excited for that collaboration. I mean, obviously, you know, in the Lyme disease world, diagnostics are one of the most challenging aspects of this disease because people have trouble figuring out they even have it. Once they figure out they have Lyme disease, then they enter into sometimes a challenging world with therapeutics where the therapeutics don't work because it's not the right medicine for the right pathogen, because it's not always Lyme disease. There are so many tick-borne diseases, or the Borrelia spirochetes have already gone deep into the system. And so someone that has late-stage Lyme, there's a 10 to 20% chance that the antibiotics don't work even in an acute case. So the late stage people really struggle. But the first step of that is obviously the problem with diagnostics. Can you speak a little bit about what you know, some of the challenges and the current state of Lyme diagnostics to be like now that it's coronavirus? I think your everyday person at the store is talking about antibody tests. So we're seeing this worldwide learning about diagnostic tests, which is something in the Lyme disease world we've been talking about for 50 years.
Yeah, you're absolutely right. The diagnostics of Lyme disease is very challenging. The only validated test in the US is a two-tier serology system, which incorporates the ELISA, more sensitive, and Western blot, more specific test. The other tests that are available out there, they're not validated, so they cannot be used in the US. They include PCR tests, antigen detection tests. Serology, as the name implies, it detects the antibodies. It takes time for a human being to develop these antibodies, so you have to wait until you can detect these antibodies, and the sensitivity and specificity of this test is not great, actually low. That's a challenge. So the approach we are trying to do is a direct test. It's not indirect as antibody-based test, it's a direct test, but it's too preliminary to say too much about the approach that we're trying to explore. It shows some promise. We are excited that we got this grant, and it will allow us to actually explore the approach that the direct detection test in diagnosis of Lyme disease.
Well, that's the point of the Emerging Leader Award. It's A, to bring in new scientists from other fields. So it's not always people who have been studying Lyme who get the award. And B, it's to try new things, give innovation a chance, because for the last 40 years, we haven't progressed a lot in this field. So I think innovation is key, and it's wonderful that you're trying something new, and we really look forward to seeing what you do with it. And I'm wondering, have you guys done other Lyme disease research in your lab previously?
Oh, yeah. One of the main focuses of my lab is to develop vaccine, subunit vaccine. We started this right off when I joined A&M University. We generated quite a lot of preliminary data, and we're trying to actually identify the potential target subunit vaccine. We're trying to be a little bit more ambitious. We want that subunit vaccine not to be only efficacious at preventing the infection, but also at having to have this therapeutic effect. So we want that vaccine to induce antibodies that would take care of those spirochetes that already got in the bloodstream. That is visible during the early stage of infection when there is a spirochetemia. The spirochetes floating in the bloodstream and they're freely accessible to antibodies. But we're doing this in the context of VLC molecule that is being expressed a few days after the spirochetes get into the bloodstream. And that VLC molecule is highly changeable, antigenically changeable, and protects, so to speak, the rest of the antigens on the surface of the spirochetes. So we're trying to find those spots that are still remain accessible to antibodies in the presence of this highly variable molecule. So,
right. Okay, great. That's a big challenge. I know people have been trying to work on vaccines for many years. There was one back in the 80s, Limerix, and it was pulled from the market. So it's a tough field. I wanted to ask you how you learned about the Bay Area Lyme Foundation Emerging Leader Award.
I think every researcher who's in Lyme disease field knows about that as a funding agency. It's a great opportunity. So I knew this when I still was in training at Washington State University, working with Lyme disease. So in my mind all the time. So
that's fabulous. Well, it's great to hear. So how did you come up with this idea for the proposal you submitted to win the Emerging Leader Award grant this year?
So I was at the birthday party. A friend of mine who's a very established, highly successful professor here at A&M University. And he invited my future to be collaborator. And when you're relaxed, when no stress, you talk about ideas. And I wanted to see what my collaborator is doing. And I told him what I'm doing. And we come up with this idea.
Did your collaborator have any personal experience or any scientific experience with Borrelia or ticks?
He had no idea what the Lyme disease, what Borrelia burgdorferi is. So he's not a microbiologist by training. He is a biophysicist. So he didn't know, he wasn't aware of this problem. So I started talking, he started talking, and then we come up with this idea.
When you heard what he did, you just thought, oh, that could be a diagnostic, that has potential.
Yes, so he's working on that tool that hasn't been yet applied to human or animal diseases, infectious diseases. So I decided, let's try it. I think it was good that we were both invited for the birthday party. So.
It seems like that's how a lot of the startups happen, right?
That's
Over here in Silicon Valley, a lot of times I hear that these kind of deals happen when people are out kite surfing.
That's right.
And the people who you work with at Texas A&M were supportive of this kind of innovative collaboration.
Yeah, absolutely. So they actually encourage you to collaborate, multidisciplinary. So it's a perfect opportunity. He has his own expertise. I have Borrelia expertise. So we just found each other on this topic, and we're trying to develop further.
And so the $100,000 Emerging Leader Award grant will enable you to do some more preliminary research on this, and then you would be eligible to apply for a larger grant with either the Bay Area Lyme Foundation does sometimes further fund our Emerging Leader Awards, or you could go to the NIH, you could go somewhere else with your preliminary data. Is that
absolutely. Yeah, you're absolutely right. So this award, Emerging Leader Award, will allow us to generate enough data because it's only at an embryonic stage of research. And so it's going to help us to explore more about this tool and feasibility of using this as a diagnostic test. And that will bring more funds later in the future, either from NIH or NSF. So, yeah.
Would you mind telling me a little bit about the human immune response to Borrelia and how the antibodies try to work to combat a Lyme infection?
Yes, the mechanism that the Lyme disease spirochetes are using to evade the human immune response. So spirochetes are considered extracellular microorganisms. So the main arm of immune system that fights off the infection is antibodies. But Borrelia spirochetes expresses VLS-E protein on its surface, and this protein is antigenically variable, so it changes all the time. So when the antibody is developed against one version of this VLC protein, the majority of the spirochete population dies off, but then there is a portion of a population that has another version of this protein, and that protein allows to protect the other antigens on the surface. So VLC has been well studied in the mouse system, and it showed that the expression of that protein on the surface is around day 3 post-infection, and that protein is essential. So if you knock it out genetically, for instance, there is no VLC on the surface, the spirochete is going to be cleared fast by the antibody response. So the VLC is very critical for the spirochetes to establish, so to speak, persistent infection in the face of the antibody responses.
And you were saying that at the beginning of the infection, the Borrelia are tending to hide. And where are they hiding?
Yes, the spirochetes don't like, the Lyme disease spirochetes don't like to hang around in the bloodstream too long, although they have this VLC molecule there. So they migrate to the different tissues, usually collagenous tissues. In mouse model, it's going to be ears, pinna, bladder, joints, heart. And that's when they try to stay there. So this way, the access of these anti-Borrelia antibodies is much restricted. So that's how they establish this persistent state of infection in the mammalian host. Yes.
So if you gave a mouse antibiotics and the Borrelia bacteria had already migrated into the more collagenous areas, then the antibiotics might not have a great chance of reaching the bacteria and eliminating them.
And that's exactly right. Yes, the more collagenous the tissues is, the more avascular. So fewer vasculature is present there. So it's way harder for antibodies to get to the spirochetes who hides in these collagenous tissues. Yes.
Right. And so I think that could be one reason why people don't always get well with treatment. It seems like these organisms, these Borrelia bacteria, are ancient. I mean, they evolved before we did, and they have pretty well adapted mechanisms of survival.
Yes, exactly. You're exactly right. And you should also keep in mind that this spirochetes, you can't find them outside the host. So they're either in the Ixodes ticks or they're in the mammalian host. And one of the main mammalian reservoir in the United States is small mice, Permiscus leucopus mice. So, and interestingly enough, to mention that if you infect these Permiscus mice or infected Permiscus mice, they don't show any signs of disease, although the tissue is full of these spirochetes.
So they've evolved to be very competent hosts for these vectors.
Exactly. Yes, that's what makes them competent, and that's what makes this life cycle of Borrelia burgdorferi perpetuate in nature. Yes, between the Ixodes ticks and Permiscus leucopus mice. Yes, and humans are considered incidental hosts.
Because in humans, I feel like I hear about people having Lyme disease, and it seems to weaken the immune system and almost make them more susceptible to co-infections. But
yes,
I know ticks can transmit multiple infections at once.
Ticks are as well co-infected with multiple genus species, or if it's in Europe, or strains, if you will, or I would say isolates if it's in the field. So, and not only Borrelia, but other tick-borne pathogens like Anaplasma, Babesia, and others. So there is a lot of co-infection going on in nature, and humans do get co-infected with different pathogens or genus species.
Well, good luck and stay safe over there. And thank you so much for joining me today. And we look forward to catching up and seeing your preliminary data soon.
Thank you so much, Leah. Stay safe as well.
Thank you for joining us for this episode of the Ticktective podcast and video series, a program of the Bay Area Lyme Foundation. You can find out more or donate at bayarealyme.org.